Hey guys, welcome back to the lecture series on biological classification. In this video, we'll talk about kingdom Protista. Kingdom Protista includes organisms which are unicellular eukaryotes. They can be categorized into chrysophytes, dinoflagellates, euglenoids, slime molds, and protozoans. Protistants are mainly aquatic, but they can also be found in other habitats as well. This kingdom forms a link with other kingdoms which deals with plants, animals, and fungi. Being eukaryotic in nature, the cell contains well-defined nucleus and other double membrane bound organelles, including flagella and cilia in some members. They can reproduce asexually as well as sexually both. Diatoms and desmids are included in chrysophytes. They are aquatic, microscopic organisms which float passively in water currents, constituting plankton. Since most of the members are photosynthetic, they are considered chief producers in the ocean. They are characterized by presence of a golden brown pigment called diatomin, which is combination of chlorophyll, xanthophyll, and carotene. Diatom have cell wall made up of two thin overlapping shells, epitheca and hypotheca. The cell wall is transparent and embedded with silica. It is fragile but doesn't decay even when the protoplasm dies off. Therefore, the cell wall is considered indestructible. Over the billions of years, diatoms have left behind a lot of cell wall deposit in their habitat. This is considered as diatomaceous earth. Diatomaceous earth is gritty and therefore can be used in polishing, filtration of oils and syrups. Some common examples of chrysophytes are Pinnullaria, Cymbella, and Melocera. This is an image of Pinnullaria, and here is the cell structure of Pinnullaria that shows two layers of cell wall, the epitheca and the hypotheca. Apart from this, it contains the cytoplasm, a single nucleus, and other cellular organelles. Hypotheca being lowermost, it is, they completely fit into each other like a soap box. As you can see here in this image, the epitheca is the topmost layer, the hypotheca is the lowermost layer, and they fit in like a soap box. Dinoflagellates are characterized by presence of two flagella. When present, one lies longitudinally and the other is transversely placed in a furrow between the wall plates, which are made up of cellulose. If immobile, the dinoflagellate will reproduce through flagellated spores known as zoospores. So presence of flagella in any life stage is a must for dinoflagellates. These organisms are mostly marine and photosynthetic. They exhibit variety of colors that depend upon the main pigment present in their cell. Some dinoflagellates are also saprophytic and some are endosymbionts. Often, a red dinoflagellate is responsible for red tides, which is due to their rapid multiplication that makes the sea appear red. Toxins secreted by these red dinoflagellates can kill a lot of other marine animals. Do you know the name of this red dinoflagellate? Comment below. Some common dinoflagellates are Serratium, Noctiluca, and Glenodium. This is the scanning electron micrograph and the schematic view of dinoflagellates. As you can see, there are two flagella. One is longitudinal and one is transversely placed there in the furrow, which is the furrow between two cell wall plates, the epitheca and the hypotheca. Euglenoids are the best known group of flagellates. They contain two flagella. One is long. I am trying to outline this for you. And the short flagella is not visible. They prefer fresh water, mostly the stagnant water. Instead of cell wall, they have a protein-rich layer known as pellicle, which makes the body flexible. They behave like a photosynthetic organism when provided sunlight. When they are deprived of sunlight, they start behaving like heterotrophs. the pigments they contain, which appears green in color here, they are similar to those found in higher plants. That is the chlorophyll A. Now, euglena is the most common example and the most studied example of euglenoids. It has a flask-shaped gullet. It can ingest solid food. In the vicinity of gullet, there is a red eye spot, which is light sensitive. They reproduce asexually by binary fission. Slime molds are saprophytes. They favor cool and moist habitat. Their body moves along with the decaying twigs and engulf organic material. When the conditions are suitable, they form an aggregation called plasmodium. This plasmodium, mind it, is different from the plasmodium vivax and malari, the protozoan, which is supposed to cause malaria. Here, plasmodium means a multinucleate protoplasm which may grow and spread over several feet. But when the conditions are unfavorable, the plasmodium differentiates and forms fruiting bodies which bear spores at their tips. The spores are meant for dispersion, so they contain true wall which are extremely resistant, and they can survive for many years until the adverse conditions are altered, and with the onset of favorable condition they will germinate once again. Some common examples of slime molds are Staminitis, Didium, Dictyostelium, Discodium. This is the life cycle of slime mold. As you can see here, the feeding plasmodium, which moves along with the twigs and leaves, it engulfs the organic matter, grows multiple times, and here the mature plasmodium, which is the multinucleated free-flowing mass of protoplasm, it grows up to several feet, but if at all the conditions are unfavorable, it may undergo making sclerotia or for spore formation. When spore formation takes place, the sporangium undergoes meiosis in order to restore haploid condition. The haploid spores will germinate, will form the amoeboid cells, or they may make flagellated gametes. In either case, the fusion of two amoeboid cells or the two gametes will produce zygote, the diploid structure, which will again start making the feeding plasmodium. The last class in our discussion is for protozoans, which are all heterotrophs and they are majorly predators or parasites. The most important group under kingdom Protista because they are primitive relatives of animals and also because they cause a lot of human diseases. There are four major groups in protozoans. Amoeboid protozoans use pseudopodia or false feet for locomotion. Pseudopodia provides simplest mode of animal locomotion called amoeboid movement. Older pseudopodia appears as wrinkles on the body called uroid. There are many freshwater, marine, and terrestrial forms. Marine forms have silica shells embedded on the surface. Some of the amoeboid protozoans are parasites, like Entamoeba histolytica that causes dysentery or amoebiasis, and Entamoeba gingivalis that is found near the mouth, in the mouth near the base of teeth. Next category is of flagellated protozoans. They have flagella for locomotion and they are either free-living or parasitic. The parasitic forms of trypanosoma causes several diseases in vertebrates collectively known as trypanosomiasis. The two important reference provided here are sleeping sickness caused by Trypanosoma gambians using tsetse fly as vector and Chagas disease by Trypanosoma cruzi using triatomid bugs as vector. Likewise, Leishmania species also cause several diseases in human and domestic animals. They are also collectively known as Leishmaniasis. Leishmania donovani causes kala-azar, the visceral leishmaniasis in humans. Leishmania tropica causes oriental sore or cutaneous leishmaniasis. Leishmania brasiliensis causes buboes or espondia, which is technically nasooral or cutaneous mucocutaneous leishmaniasis. This is Trypanosoma brucei, the schematic diagram of Trypanosoma brucei. Here is the free flagellum. Here the flagellum is attached to the body. This is the undulating membrane. We can see the other cellular structures and the presence of Trypanosoma in blood. These are RBCs. And we can see the Trypanosoma brucei acting as a blood pathogen. Next category is of ciliated protozoans, which definitely use thousands of cilia for locomotion. They are aquatic, actively moving organisms and have a cavity or gullet just like euglenoids that opens to the outside of the cell surface. The coordinated movement of rows of cilia causes the water-laden food to be steered into the gullet. And this is how paramecium feeds on the food available in water. Paramecium is slipper-shaped. The lower body of the surface has oral groove, which helps in ingestion of food. Like amoeba, it also has contractile vacuole for osmoregulation. Asexual reproduction is by binary fission. However, sexually, they reproduce by conjugation. This is an electron micrograph of paramecium, and here is the schematic diagram of paramecium showing the oral groove, the gullet, the cytostome. We can see two nucleus, macro as well as micronucleus. There are contractile vacuole, the anterior one and the posterior one. We also have thousands of cilia over the surface of body, and the cytoplasm is divided into ecto and endoplasm. Ectoplasm towards the outer surface and endoplasm towards the inner surface. The last group of protozoans are sporozoans that include diverse organisms having an infectious spore-like stage in their life cycle. Of all the sporozoans, the most notorious is Plasmodium, commonly known as malarial parasite, the causative agent of malaria. Malaria is characterized by malaise, fever, chills, sweating, headache, and nausea. Malaria is known to be caused by 4 species of genus Plasmodium. There are more, these are the most common ones. Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae, and Plasmodium ovale. All of them are intra-erythrocytic sporozoan parasites, means that they live within the erythrocytes or the RBCs. Worldwide, Vivax malaria is the most common type of malaria. However, falciparum malaria is the most deadly, and the least common one is Plasmodium ovale malaria. This image, you can see here different stages of malarial parasite in human bloodstream. In the next image, this represents life cycle of malarial parasite in both the host. It starts with the mosquito biting a human, and through the mosquito bite, sporozoites are injected into human bloodstream. The sporozoites invade liver cells, then gets converted to hypnozoites, the dormant stage, then into schizons, and now merozoites are released from schizons, which invade the RBCs. They get converted to trophozoites and then to schizons and then to merozoites again in the RBCs. Merozoites develop into male and female gametocytes in the RBCs only. At this point, when the mosquito bites human again, the gametocytes enter into the mosquito's stomach, wherein they fuse with each other. The gametes are produced. Gametes will fuse with each other, producing zygote in the mosquito's stomach itself. Zygote gets developed into ookinete and then into oocyst. Oocyst develops on the outer wall of stomach. Sporoblasts develop within oocyst, and sporozoites are released from the oocyst, which enters the salivary gland, and the cycle continues when the mosquito bites human again. That's all in this video. In next video, we'll talk about kingdom fungi. Thank you. Stay tuned and please do subscribe our channel.